Hi, everybody. Okay, now we're moving on to chapter 3. Okay. Screen. Okay, so hopefully these 3 chapters, chapter 1, 2, and 3, are more of a review for you guys in actual physiology and learning new material. Okay. Okay. So in anatomy, we learn how to subdivide compartments and stuff. Okay, and that's based on physical structures. Okay, in physio, we'll do it on more function structures. Okay, and you'll see that this chapter in its entirety is a rehash of parts of anatomy, but with relation to function rather than anatomical features. Okay. Okay. So the 3 major cavities of the body. Okay. The posterior cavity is basically called the cranial cavity. We don't really deal with the spinal cord very much. So the cranial cavity. The ventral body cavity, the one in front, is actually subdivided into 2, the thoracic cavity and the abdominal pelvic, because they don't really interact with each other very much. Okay, so thoracic cavity on top, abdominal pelvic on the bottom. Okay, and then you have different fluid compartments, the circulatory system, the eyes, the cerebrospinal fluid, and then the pleural and pericardial sacs. Okay. Okay, so there's body cavities. Okay. 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 So one of the hardest things for students to really envision, and if you guys told me with anatomy, I made sure you understood that, is that the lumens of some organs are actually outside the body. So, like the best example is your stomach. Okay, the inside of your stomach is actually considered the outside of the body because it's not actually absorbed. Like the food that's inside your stomach is not really absorbed into you until it gets past the stomach into your blood vessels and then from your blood vessels to your cells. Okay, the word lumen means light. Okay, if you're cutting slides and stuff, you're making slides, there's where there's no tissue, there's light coming through. So the lumen is the interior of any hollow organ. Okay, it's usually filled with air or fluid of some sort. Okay, for most organs is actually an extension of the external environment. Okay, so heart, lungs, blood vessels, intestines. Okay, all right. Functionally, the body has 3 fluid compartments. Okay, the intracellular fluid, everything inside the cell. Okay, and then the interstitial fluid. Okay, and then the plasma. Okay, the plasma and interstitial fluid has less difference between them than either one has intercellular fluid, but the plasma has a whole bunch of big, huge proteins that do not cross the leaves of blood vessels, so they don't actually go into the interstitial fluid. Okay. Okay. So here's the blood. Okay, and interstitial fluid. So the blood plasma, in addition to having the blood cells, actually has a bunch of blood proteins that are really big and don't cross this boundary. Okay. Okay. So this is the interstitial fluid. Inside the cell has its own fluid, the ICF or intercellular fluid. Okay. All right. anytime you want a compartment inside a body, every compartment has a membrane around it. Okay, there has to be some way of separating that compartment. Okay, so whether it's at a visible level or microscopic level or even a electron microscopic level, you have boundaries. These boundaries are always separated by membranes. Okay. Okay. All biological membranes have some features that they share in common. One, the fact there's a membrane, physical membrane, okay, allows for physical isolation. It separates the inside from the outside. Okay. In addition to separating the inside from the outside, now that I have a way to separate the inside from the outside, I have a way to regulate what can come in and what can go out. Okay, and that's the purpose of the membrane. The regulation of the exchange with the environment. I can let things in if I want to. I can prevent them from coming in if I want to. I can allow things to stay in if I want it to stay in, or I can let it out, or I can even push it out on purpose. Okay. Okay. In addition to regulation, there's communication between the cell and its environment. So I know what's on the outside and I know what's on the inside, so I can regulate which way they go. Okay. And then last but not least, the structural support, the physical purpose of having a membrane helps hold the cell in. Okay, and then secretion, it allows certain things to be pushed out, right? So most membranes are lipid and protein, okay? And those lipid and protein work in something like a fluid mosaic model, okay? You can go over that in class. I still understand what that is. Okay. But just because it's lipid and protein doesn't mean all the membranes are the same. Okay. Some membranes of your regular cells, okay, are actually more lipid, less protein. Here they compare 3. Red blood cells have about 50-50 really. Okay. The myelin, because myelin needs to insulate neurons. Okay, if you guys remember from anatomy what myelin does, it needs to insulate neurons, so it has to be mostly fat. Okay. On the other hand, the internal mitochondrial membrane has a lot of molecular function. Like there's a lot of stuff that's going on the surface of the membrane. Therefore, the surface of the membrane has to have mostly protein, because protein is where all the chemical reactions are happening. Okay. Okay. So this is the fluid mosaic model. Okay. So this is your normal cell, right? This is what your normal cell really looks like. It has mostly phospholipids with proteins that are sticking in and out of it. Okay. Okay. There are a couple of different types of protein. Okay. There are, let's see, I'll make sure you guys understand. Okay. So integral versus peripheral. Integral means through the membrane, and they're always transmembrane proteins. Okay. So they cross the membrane. So this is an integral protein. Okay. This is a transmembrane. So you can see there's a lot of proteins that cross the membrane 7 times actually. So if you count 1, 2, 3, 4, 5, 6, 7, like there's there's 3 coils behind these strings. So 1, 2, 3, 4, 5, 6, 7. Okay, they're called 7 span membrane proteins. Okay. Okay. All right. What else? Oh, peripheral proteins. Peripheral proteins are either on the inside layer or on the outside layer, but one peripheral protein is never on both sides. Okay, so they're on one side of the membrane or the other, and they can, cause different reactions to happen on one side or the other. Okay. Okay. What else is there? Okay, lipids. Okay. Here are the membrane phospholipids. Okay. So this is a typical picture of a phospholipid. If you put these two together, if you put this together, okay, they can form a couple of different structures depending on how much there is. Okay, if you just have a little bit of this together, then this is your most stable formation. Okay, this little ball where all the hydrophobic things are on the outside. Sorry, all the hydrophobic things are on the inside. All the hydrophilic things are on the outside. And these are little micelles. These are important in terms of lipid digestion. This is how you carry around oils and stuff. Okay. More stuff. If you have more stuff, then you form this. Okay. Or you form a small little bilayer and you have an aqueous center. You form a bigger one and you get something that looks like that, a phospholipid layer that forms a sheet. And that sheet can actually look, basically, your cell membrane is a phospholipid bilayer. Okay. Cool fun fact: the RNA vaccine you guys probably got, like the Pfizer, Moderna, are actually coated in liposomes. That's how they get into your cells. Okay. So lipid rafts. Okay. Okay. So make sure you kind of understand all this stuff. Okay. It's a concept map. Okay. Don't worry about lipid rafts. They won't be on the test, but these are lipid rafts. Okay. So the whole concept of the fluid mosaic model is that these things are mobile and they're constant. These rafts are kind of floating around on your cell membrane and they're kind of moving across your cell membrane slowly, like rafts would do. Okay. Okay, glycocalyx. If you're taking microbiology, you understand how important glycocalyx is in bacteria. Okay. It's just on an external surface surrounded by glycolipids and glycoproteins. So you have all these sugar molecules on the surface of the cell. Okay. Okay. Again, if you're taking microbiology, you would know that eukaryotic cells are bigger than bacterial cells, and that's because eukaryotic cells have internal compartments. Okay, and these internal compartments allow these different compartments to do certain things, and only those things. Okay, so you're dividing up, an office into small little rooms where different rooms will look slightly different and have different functions. Okay. So here is the cellular structure of a eukaryotic cell. So again, we're focusing on human physiology. So human physiology, we're going to be looking at what happens in a eukaryotic cell. Okay, so there is cytoplasm, okay, which is divided into cytosol, which is the fluid, and then the organelles. All of these organelles have their own function, okay, and then the inclusions are the inclusions are basically storage structures, and then the protein fibers are just proteins. Okay, so inclusions are direct contact with the cytosol. So this is how we store fat and store glycogen. And then ribosomes sometimes are included in inclusions, sometimes they're included in organelles. But ribosomes are made from RNA and protein, and they manufacture proteins. Okay. Okay. So cytoplasmic protein fibers come in 3 different sizes. The smallest ones are actin fibers. The bigger ones are the intermittent filaments. Okay. The microfilaments allow the cells to move. The intermediate filaments allow for stabilization of the cell shape. Okay. It prevents squishing. Okay. And then microtubules are like also allows the cell to move the bigger components, and it also counts as the freeway system inside the cell. So when things move inside the cell, they don't just float around. They actually move around through these microtubules. Okay. So there you go. Okay. You can read all these guys. Okay. There's not much of this. Okay. Microtubules form centrioles, cilia, and flagella. Again, this is a slide that you should probably already know from anatomy and stuff like that. So, okay. All right. Okay, it's measurable, blah, blah. Okay. Okay, so cytoskeleton is a changeable scaffold. So cells are actually extremely dynamic. And I don't think we explained it really good, and definitely not in high school biology. And you guys probably don't have enough biology background, but cells are extremely dynamic, change constantly. The internal environment changes constantly, and therefore the cytoskeleton is not solid in any way. It's like a scaffold that dynamically rearranges itself every time you need to do something. Okay, so this shape of the cell, if it needs to bend or move, the cytoskeleton can change shape. The internal organization, where the mitochondria are, and where, like, how big it is, and all this other stuff, okay, allows movement. Okay, intracellular transport using these motor proteins. So here's motor protein. You can actually hook inclusion or organelle onto a motor protein, and it will literally walk down the cytoskeleton. These are the microtubules actually, and it'll actually walk down there. So things are actually carried down the cytoskeleton fibers. Okay, to give you a really dramatic example of this, humans have neurons that are over 1 meter in length, so like 3 feet. Okay, and bigger people almost 4 feet long. Okay, and these organelles have to be carried from one end of the axon all the way down to the other end. So they really have to walk motor proteins. They really have to walk down the cytoskeleton that way. Okay, and it takes a long time to actually get down there, but they move slowly down there. Okay. All right. So organelles create compartments for specialized functions. Okay. So these guys I won't go over because we already went over them in anatomy probably. Okay. So you just look at those. Okay. Or if you haven't, then you learn them by yourself. They're not difficult. Okay. Okay. The nucleus is where your DNA is kept. Okay. Your DNA is not naked. It is wrapped up in protein to prevent things from happening to it. And therefore, it's usually correctly called chromatin. Chromatin is just the DNA and the wrapping of proteins around it. Okay. The DNA and protein, the chromatin, is actually surrounded by a nuclear envelope. Okay, so an envelope is another lipid layer, bilayer. Okay, but this one is much tighter and doesn't allow anything through. Okay, it's like a security fence. It doesn't allow anything through. The only way through are through the gates. The gates in this case are the nuclear pores. Okay, the nuclear pore complexes is what determines whether something can come in or out. Okay, so if you're a big molecule, the only way you come in and out of the nucleus is through one of the nuclear pores. Okay. Okay. Nucleus. Okay. So a good way of understanding how the subcompartments actually work is you go through this and you see how many, like to make something come out of the cell, look at how many compartments you have to go through. One here, one here, one here, one here, one there, one there. So you have to go through each compartment, and each compartment does something different. Okay, and you can read all that yourself. Okay, so we'll stop right here. Okay, in this recording and in the next recording, we'll talk more about tissues. Okay.